everyone. So I'm Miss Ray, and today this is the recording that goes with the live practice for the SOL Scientific Method Week 1 work. So we're going to be talking about scientific investigation and a lot of the different important aspects that we learned about in Unit 1 that are going to be most important for your SOL test. As we go through, I'm going to make sure to tell you several memory strategies and vocab relationships that'll help you remember some of this information. Alright, so first we're going to start with the differences between observations and inferences. So observations are going to be very important in science because they're the basis for everything and the way that we act and react to the world around us. So observations are information, or they're made and gathered using your 5 senses. So you either see it, hear it, taste it, touch it, or smell it. So when you look around and you see that the grass is green, the sky is blue, there are 2 dogs, those are observations. And observations can be one of 2 types. They are either qualitative or quantitative. So observations are either qualitative or quantitative. Qualitative observations are based on appearance. This could be color. It could be texture, whether something's wet or dry. So when you walk into your biology room and you see that the walls are green, that the tables are black, that the counter is rough, or the rock is smooth. These are qualitative observations. A quantitative observation is going to involve a number, and I like to remember that that's what this N stands for, is number. So any observation that you make that involves a number is quantitative. There are 5 students in the classroom. There are 14 tables. The student walked to the trash can 2 times. Any observation that involves a number is quantitative. And we use our observations to then make inferences. An inference is a guess based on our observations. Now, just human tendency, oftentimes you skip right to your inferences and you don't even really think about your observations. For instance, you're walking down the road or something and a neighbor's dog barks at you. You might just automatically think that, oh wow, that's a mean dog. But you don't know that. The reason that you made that guess or that inference is because maybe to you it just sounded a little bit more aggressive. But I'm sure that the owner might know that it could be a very loving dog that's just, protective and barks at everything that goes by. I have one of those dogs myself. And in reality, he sounds, vicious when you walk by, but he's really not. He wouldn't bite anything. And same can be made for this picture here in this image. You see a boat in a pond with a young boy. You might automatically assume that the boat belongs to the boy, but you don't know who the boat belongs to. That could be a little boat that just had been left in the lake. Maybe a friend let the boy borrow the boat. You don't know. You don't know that it belonged to the boy. It's just a guess. That's just an inference. Another important term in biology is the term theory. A theory is something that's created when a hypothesis has been tested many, many times and seems to be true. So it's supported by lots of evidence. An example of this is the theory of evolution. There are many pieces of evidence that support the idea that evolution is true. That doesn't mean that something can't be found tomorrow that disproves evolution. It just means that we have lots of evidence, many things have been done to suggest that the theory of evolution is true. So theories are supported by lots of evidence and lots of experimentation. Moving on to the scientific method. The scientific method has 5 general steps. As we go through, I'm going to give you some examples. So the first step it could be stated in many ways. So the first is to state the problem. This comes from you making an observation. It could also be phrased as a question. You make an observation and it leads you to a question. So an example, I grow lots of plants at my house. I have like tomato plants and pepper plants, and I like planting a garden. So I'm growing lots of plants right now. I might have a question of, I know how plants grow in normal sunlight. I wonder how they would grow under different color lights. So that's my problem, my question. How will my plants grow in different colors of light? From there, I form a hypothesis. And your hypothesis is your guess of the answer to the problem. So this is your guess about what you think is true. From there, you create a prediction. Now, I'm sure you're thinking, Ms. Ray, hypothesis, prediction, those are like the same thing. And they're very close. The difference between a hypothesis and a prediction is that a prediction is worded if I am correct, then this is what it will look like. So if I am correct, then this is what it will look like. So my prediction for my problem, let's say my hypothesis is, I think my plants will go best under blue light. Then my prediction would be, if plants grow best under blue light, then the plants under blue light will be taller. Next, you perform your experiment. We'll get into all the different steps that go into that here in a second. And then after your experiment, you have your data. You draw a conclusion. Now, your experiments, they're not just simply something you toss together. In order to have a valid experiment, they have to have a few different parts. The first part of a valid experiment is the independent variable. The independent variable is what I, the scientist, change in an experiment, what the scientist manipulates. Manipulates just means changes. So in my experiment, I would be changing the color of light. I might have some in normal sunlight, I might have some in blue light, some in red light, some in green light, etc. And then the dependent variable will change depending on the independent variable. And the way that I like to define this is what you measure in the end. So in my experiment, in the end, I measure the height of the plant. My dependent variable is the height of the plant. So in my experiment, I have different groups. I have several groups of plants. Plants in normal light, plants in green light, plants in blue light, plants in red light. Those are all my different groups. And those groups are going to be put in 1 to 2 categories. They're either going to be experimental groups or control groups. Experimental groups get the new thing that you are testing. They get the IV. The IV is the new thing that you are testing. In my experiment, the new thing I'm testing is like blue light, green light, and red light. I don't know what happens to a plant in those different colors of light. That's not normal. So that's my experimental group. They get the new thing that I'm testing. Your control group is also known as your compare group, because you're going to compare back to this group, and this gets the normal thing. So in my experiment, the plants that are in normal sunlight are the ones that are my control group, because I know what happens to plants in normal sunlight. It's normal for plants to be in normal sunlight. That's my control group, and I'm going to compare the groups that get the different colors of light back to the normal light. Now, you do not want to get this confused with a constant. Constants are factors that stay the same to make your experiment fair. So, for example, if I'm changing the colors of light, then I need to use the same type of plant, the same amount of water, the same amount of time in the light. I need to keep every single thing the same except the color of light, because that's what I'm testing. It's not fair to put a fast-growing plant and a slow-growing plant in the same experiment if I'm testing colors of light. If I'm testing colors of light, that's the only thing that changes between the groups. So I have plants in normal light, red light, green light, blue light. They're all tomato plants. They all get watered twice a day. They all are in the sun for 8 hours a day. Everything else is the same, except for the colors of light. And when you get questions on your SOL test about what makes an experiment not valid, if they do not have a control group, it's not a good experiment. If they change more than one thing, it's not a good experiment. So these all have to be in place for this to be a quality experiment. All right, moving on to graphing. The main thing to know about graphs on the SOL, you will have to analyze them, but you do need to know that the independent variable is always on the x-axis, and that the dependent variable is always on the y-axis. So in this case, the independent variable is time because it's on the x, and the dependent variable is temperature that's on the y. We will have practice analyzing graphs, so make sure you check that out as well. Alrighty, for microscopes, one thing you might need for microscopes is the total magnification equation. The total magnification of microscope is equal to the eyepiece, which is always 10x unless you're told otherwise, times the objective lens. Now you'll either be solving for the total magnification or the objective lens. Remember, if you're solving for the total magnification, you will be multiplying 10 times the objective. If you are solving for the objective, you're going to be dividing total magnification by 10 to get your objective lens. Alrighty, and then one of the last, but certainly not least, because this is very, very important for this unit, are the 5 characteristics of life. All living things have 5 things in common. First of which, they have cellular organization. They are made of cells. All living things are made of cells. It's part of the cell theory. They also have to be able to maintain homeostasis. Homeostasis is balance, stability, ability to stay the same, to maintain an internal condition. If your question is asking about homeostasis, or the answer is homeostasis, you're going to have the word balance, maintain, internal condition, stable, stasis, stable. These are your key words for homeostasis. All living things also have metabolism. Metabolism are all of the chemical reactions that occur in your body. Your key words for metabolism are chemical reactions and energy. If it's asking about metabolism, you're going to have one of those words in your answer or in the question, depending on what it's asking. All living things have DNA, and this leads to heredity. The ability to pass your traits on to your offspring. So passing down of traits. Parents look like children, children look like parents, so on and so forth. And the last characteristic is all living things can reproduce. Some living things reproduce asexually, like bacteria. Some living things produce sexually. They might reproduce differently, but they can all reproduce. All living things have these 5 characteristics. Make sure you pay close attention to the different keywords that will lead you to your answer. So please make note of these keywords. Another important keyword to note over here, control group, compare group always go together. Keywords are very, very important when you're preparing for your SOL. All right, if you have questions about any of this, please reach out to your teacher. Thank you.